Okay, so I'd just like to start off by thanking the organizers and all of you for having me here to speak today, and by acknowledging my co-authors for the work I'll be discussing. Jenny Boston, who of course is the organizer of the symposium, and Don McLeod. So just briefly, anomalous trichromacy, as we all know, is a type of color vision deficiency where there are three types of cone photoreceptors, like in normal trichromacy, but two of the cone types are more similar in spectral sensitivity. And so this decrease in spectral separation, of course, results in a reduced ability to discriminate between colors that are reddish and greenish. But when we think about what the world actually looks like to someone with anomalous color vision, things are a bit complicated. So one possibility, of course, is that the similarity in cone sensitivities leads to a reduction in cone contrast that's applied to the full red-green dimension of color space. And so as we can see here in this figure from Webster and colleagues, which is a simulation of how the chromatic contrast of an image might be reduced in this way. But an important implication of this is that the red-green opponent system would be understimulated by the anomalous cones, and therefore it wouldn't be making use of the full neural capacity or dynamic range of the opponent neurons that encode chromatic contrast. So another possibility is that neural compensation boosts the photoreceptor signals to make use of the full neural output range. So this would mean that their color perception is actually much more similar to normal than what would be predicted simply by their cone sensitivities. So Regan and Mullen first suggested this in 1997 when they compared the relative salience of the cardinal axes of color space in normal and anomalous trichromats, and found that for some anomalous trichromats, the relative salience was normal or near normal, but they didn't measure discrimination thresholds. So it remained possible that their results could be explained by near normal color discrimination abilities in their participants. So this idea of compensation in anomalous trichromacy was the inspiration behind the work I'll be showing you today. So we were interested in comparing the ability of anomalous trichromats to discriminate reddish and greenish colors to what the appearance was above threshold. So first we had observers perform a threshold sensitivity task using a 4-alternative forced choice procedure where subjects were asked to identify which quadrant of the stimulus shown here appeared different in chromaticity. And so the chromaticity of the test quadrant varied along the cardinal axes of the McLeod-Boynton chromaticity diagram. And as expected, we found that thresholds for S over L plus M, or the blue-yellow axis, were roughly equal for the three subject groups, but the L over L plus M axis thresholds, or red-green thresholds, were elevated for deuteranomals and protonomals relative to normal trichromats. So in the second task, we had the same observers rate on a 0 to 9 scale how different two colors appeared on each trial. And the stimuli used in this task are shown here, plotted in McLeod-Boynton space for normal trichromats. And to the right, you can see how these are plotted in anomalous chromaticity diagrams with different spectral peak separations with the DeMarco, Pokorny, and Smith anomalous cone fundamentals. So we use these difference ratings from the experiment to construct maps of perceptual color spaces with multidimensional scaling, or MDS. And so this generates a map of the colors where the distance between each point corresponds to the perceptual differences between the colors. This is a way to model perceptual color spaces. And so as you can see, the average MDS solutions here for each subject group with ellipses fit to each saturation level. What we found was that the contraction along the red-green axis was much less than expected from predictions of how these stimuli are represented in a physiological color space. So even though deuteranomals and protonomals had sensitivities of only about 40 and 20% of normal, the contraction along the red-green dimension from the color appearance ratings was only about 90 and 70% of the mean normal trichromats. And if we look at the contrast response functions, we're plotting the x-intercepts of each fitted ellipse as a function of change in L over L plus M of the stimuli. We see a much more similar to normal contrast response function for anomalous subjects than what we would predict from their elevated thresholds. So the main takeaway from these experiments was that anomalous observers had much more similar to normal MDS solutions than expected from their reduced sensitivity near threshold. This suggests that post-receptoral compensation might amplify the cone inputs. But one explanation that our study didn't really rule out is this idea of a cognitive compensation instead of a neural compensation like I've been describing so far. So for example, individuals might just learn to report that reds and greens are more different from one another based on experience. So in a second study, which isn't yet published, we tried to get at this with a more objective task than the subjective MDS color difference ratings. And so we looked at how anomalous observers' thresholds changed with chromatic contrast of a pedestal stimulus. So this is well known from previous work that pedestal contrast versus discrimination functions follow this V-shaped function with the lowest thresholds near the background chromaticity. And so this is believed to be due to the compressively nonlinear shape of the chromatic contrast response of post-receptoral neurons. So we'd expect if there were no compensation in the anomalous visual system, the neural signal representing a particular contrast would be smaller than it is for a normal trichromat. And so this would effectively stretch this V function out horizontally, since greater pedestal contrast would be needed to achieve the same response in a normal trichromat for any of these pedestals here. And as you can see in our results here, that's sort of what we see, that thresholds for anomalous observers, while they're raised relative to normal, they're also showing us that they have slopes, the slopes of the functions are reduced. So we first considered our results in the context of this type of zero compensation model, where we're considering here threshold or delta R for detection of a chromatic stimulus on a chromatic pedestal. And K here is related to the anomalous pigment sensitivity. So 1/K is the ratio of anomalous to normal cone separations. So we applied this model for several values of K ranging from 1 to 12 nanometers. And we did this because we didn't actually know the spectral separations for our anomalous participants. But we find that for any of these predictions of spectral separation, the slopes for anomalous participants are much steeper than what this zero compensation model predicts. So we next considered a model that takes into account noise prior to the compressive nonlinearity or input noise. So the predicted V profile for anomalous observers depends on the location of the predominant source of noise in the visual system, and particularly whether it occurs before or after the level of compensation. So this matrix of plots here is showing you increasing compensation from 0 to 100% horizontally and input noise vertically. So we considered input noise as a percentage of the total noise that contributes to the observer's threshold. So this panel here in the upper left corresponds to the zero compensation model I showed you on the previous slide. And if we only really consider that the source of noise occurs after compensation, so that's this row here, the full compensation model in this case would predict thresholds exactly the same as the normal trichromat. And this is because in this case, compensation occurs prior to the point of the limiting noise. But it's reasonable to assume that input noise that occurs prior to the compensation could also be significant. So ultimately, we found that all of these models considering different levels of input noise didn't really predict the data very well. And while our data sort of refute the zero compensation hypothesis, we don't really get a complete picture from these models. So to just summarize briefly, before I share just a couple more slides, while we see evidence for compensation in our studies, the simple models of compensation we applied to our pedestal discrimination data don't really account for our findings. But I'd like to take the opportunity to point you to a couple of newer studies along this line of work that have come out more recently in support of the compensation hypothesis. So first, Knobloch and his colleagues used maximum likelihood difference scaling to look at the superthreshold contrast responses in anomalous and normal trichromats. And I believe he's actually also scheduled to speak, so we might hear more on this work directly from him. But second, Katie Tregelis and others showed compensation at the cortical level using fMRI to image anomalous Gould responses to chromatic contrast. And so this was a really exciting result, and specifically they found that compensation didn't appear in area V1, but the contrast response functions were indistinguishable from normal in areas V2 and V3. So this is direct evidence of a neural compensation as opposed to just a cognitive or biased response in the anomalous observer. So it's possible that a more complex model, for example, one that allows for noise at the compressively nonlinear stage, but compensation at a later stage, like in the cortex, or even at multiple levels of the visual system, might better explain our results. But in any case, there's starting to be a good body of work supporting the idea that color appearance in anomalous trichromacy is actually much more similar to normal than we might expect. And with advances in technology like fMRI, I think some exciting work is on the horizon, which will hopefully tackle some of the remaining questions that we have. So to that end, I just want to describe briefly some of the work that we're doing at UC Berkeley, where we're using adaptive optics, or AO, which is a set of techniques that corrects the aberrations in the eye's optics and allows for retinal imaging and stimulus presentation at the resolution of single cone photoreceptors. So a lot of the work that's been done so far to deliver stabilized, optically precise stimuli to the retina was pioneered by Austin Rorda's group at Berkeley, and along with Austin and several others, notably Ren Ng and James Fong in electrical engineering and computer science, who have really been driving a lot of the software advances for this work. We've been developing a new platform for color vision experiments where the L and M cone types can be targeted independently in an adaptive optics scanning laser ophthalmoscope, or AOSLO, which is the tool that we use to image and stimulate cones in the retina. So this video I'll play for you now is showing, let's see, there we go. It's showing the cone photoreceptors as they're imaged by the AOSLO. And so this video is slowed down a bit so that hopefully it plays all right over Zoom. But normally we're imaging in real time at a frame rate of 30 hertz. And since we have knowledge of the spectral types of cone, of each cone from a procedure called densitometry, we're able to then modulate the intensity of a laser that we're using for stimulation of the cones to create what we can think of as a spatiochromatic metamer. So we're all familiar with the concept of spectral metamers, where different combinations of wavelengths and intensities can be used to generate the same cone activities and thus the same color percepts. But in this case, we're actually modulating the intensity of a single wavelength of light as we deliver microdoses to the L and M cones in order to generate chromatic percepts. So we've done a couple of preliminary experiments with stabilized stimuli to validate that these spatiochromatic stimuli actually generate chromatic percepts. So in this figure on the left, I'm showing results from a color matching experiment that we did with Brian Schmidt, who's now at iHeartRadio, where these are the CIE chromaticities of stimuli that were color matches made with an external projector system in Maxwellian view. So we also did a pedestal discrimination experiment. So you might recognize this V-shaped function is quite like those that I showed earlier. And so really the hope of this work in replicating some of the classic color experiments is that we can validate a new platform for color experiments that allows us to investigate color vision on a very fine spatial scale with knowledge of the spectral cone types and the ability to present chromatic stimuli to the observer, where we're defining each stimulus in terms of cone intensity contrast so that we can theoretically present any possible LM cone contrast to the observer. So obviously, the work I just showed you relies on having spectrally classed cone mosaics for our subjects, but so far this hasn't been possible with anomalous trichromats. And so this is because classification of cones is done with a technique called AO densitometry. And this was first done by Rorda and Williams in 1999, and it uses reflectance imaging of the cones after selective wavelength bleaching to delineate their spectral classifications. So this is a pretty difficult and time-consuming process that relies on collecting a lot of data to overcome signal to noise constraints. And for a long time, the possibility of cone classing was restricted to either normal trichromats or dichromats, with the anomalous cone pigments being too similar to really distinguish between their cone types. But just last year, Zhang and others from Don Miller's lab at Indiana used phase dynamics in OCT imaging to look at changes in the outer segments of the cones. And this methodology is a pretty big improvement in sensitivity over retinal densitometry. So as this technology continues to improve, I think there's a real hope that it'll be sensitive enough to tease apart cone types and anomalous retinas, and make it possible to do these types of psychophysical experiments that rely on spectral classifications with anomalous observers. So to conclude, I'd just like to thank all of you for your attention and also acknowledge again my collaborators on this work, Jenny Boston and Don McLeod, and the group at Berkeley, and also funding sources supporting this work. So thank you.